I was slightly unexpectedly seeing me as chair. Actually, Cal is one of my PhD students, just finished with me. So Cal is going to discuss quantifying the early cardiac transcriptome and rat models of left ventricular hypertrophy with or without predisposition to hypertension. Cara, thank you.
Thank you, Martin, for the introduction. Hopefully everyone can hear me. As Martin said, I'm a PhD student. I'm at the University of Glasgow, and I'm going to take you through some of our RNA sequencing data today that uses genetic models of hypertension and cardiovascular complications. So yeah, here we go. So rodents are great preclinical models. They can translate findings from human genetic studies, but also they can be used to investigate mechanisms. And in our research, we tend to focus on high blood pressure and the associated cardiovascular risks. And there are actually over 27 models of hypertension, and that's from a pool of over 100 in cardiovascular disease. In Glasgow, we have the stroke-prone spontaneously hypertensive rat and its closest genetic comparator, the WKY. And these have been maintained as separate colonies in Glasgow for over 20 years now. I'm sure you're probably aware, but the SHRSP, they become hypertensive from about 12 weeks of age. And stroke will occur stochastically, particularly if we apply additional stressors such as tack surgery or salt loading. But they also develop other complications that is quite prevalent in humans with hypertrophy, in hypertension, and that's left ventricular hypertrophy. Apologies. So this is independently associated with an increased risk of cardiovascular morbidity and mortality, and left ventricular mass is actually highly heritable. So left ventricular hypertrophy is an adaptive or a maladaptive increase in the left ventricle size, and it can occur in physiological stimuli such as exercise or pregnancy, but it often occurs during hypertension, and it's related to an increase in cardiovascular disease morbidity and mortality, as I said, because the heart's ability to actually pump blood around the body doesn't change with that increase in muscle size. Left ventricular mass, left ventricular hypertrophy actually occurs non-uniformly in these hypertensive patients, and that's probably due to this highly heritable component of left ventricular mass. So it's important to try and understand some of the causal pathways that underlie this remodeling. So using an F2 cross between the Glasgow WKY and SHRSP strains, we previously identified a QTL for left ventricular mass on the rat chromosome 14, and this was independent of blood pressure. So the region was selectively bred into two reciprocal congenic strains, so that's both on the WKY background and the SHRSP background, denoted by the strain coming first in their naming. So the congenic strains showed blood pressure profiles that matched their background strain, but structural and functional differences in the left ventricular in the left ventricle that developed from actually 12 weeks of age, so before the onset of higher blood pressure in the SHRSP background animals. In early microarray studies at this sort of 5 and 16 week stage of development, there was an increase in the expression of genes that were associated with cardiac hypertrophy, but these studies in 5 week and in adulthood are in an already remodeled myocardium, so they don't give us an insight really into the causal effects of the congenic region. So put simply, our question is, can we find important genes and pathways that causes this remodeling in hypertensive and normotensive animals? So we wanted to look at those potential genetic mechanisms that precede the development of dysfunction. So we looked at GD18 animals, and now we chose this time point because the heart is fully formed by this point, but it's not under the hemodynamic load and remodeling stimulus that occurs in the first 10 days of life. It's also prior to any differences in the left ventricular mass, which occur from 5 weeks, as I already said. So we aim to perform RNA-seq in this cardiac tissue from the WKY, the SHRSP, and the 2 chromosome 14 congenic strains. We also wanted to explore the role of alternative splicing in these models. And then finally, because RNA-seq experiments are often affected by the pipeline of analysis, we had this secondary aim of sort of quantifying this effect in the rat. Much of this research tends to focus more broadly on the human and the mouse. So we have 4 groups to make our comparisons between, and we tend to focus on the SHRSP to the WKY comparison, so that's the normal parental strain. Then we compare the chromosome 14 congenics to their background strain. We use a standard RNA-seq pipeline as kind of details here, and we use a ribosomal depletion in this library prep to capture both non-protein coding and coding sequences. Wrong way. We compared the pipelines of analysis that changed the quantification process here. So across the 3 pipelines, the same set of reads were read into HiSat2 or to the pseudo-alignment Callisto. And we did a gene level count using feature counts, and that's where quantification is obtained by just counting the reads overlapping each gene. And then we did a de novo transcript assembly using string tie. And as I said, the Callisto software, these two estimate transcript level counts, and we use TXI import to impute these into gene level counts that are appropriate for DSeq2 or differential gene expression, and a differential transcript usage analysis, which compares the proportion of transcripts that contribute to a gene expression and compares that between 2 groups. So we found that HiSat2 as expected produced a much higher percentage of aligned reads compared to using Callisto, but that's expected as Callisto is only aligning to the transcriptome, whereas HiSat2 aligns to the whole genome. And we used a ribosomal depletion, as I said, so we expected a higher number of reads to potentially map to intergenic and intronic reads. But also this is in cardiac tissue, which has a really high mitochondrial content, so that can also affect this ability to align to the genome. String tie assembled a transcriptome from the GD18s that had over 99% of the transcripts within it. It also annotated an additional 40,000 or so transcripts that were not annotated, so that's this precision marker here, suggesting that there might still be some room for increased depth of transcription, sort of number of transcripts per gene in the rat annotation. But again, that's not necessarily new information. So string tie and Callisto, which are these pipelines 2 and 3, seem to have a much higher sensitivity than feature counts. So when we looked at the distribution, we had a really high number of very low or non-expressed genes in these 2 pipelines. So when we removed these genes that had really low expression, we found that Callisto seemed to retain the highest number of genes for us to use into downstream analysis, and it lost a less percentage than string tie, which again was expected from that distribution of counts, but lost slightly more than feature counts. We constructed DSeq models from this filter data set to reduce noise and increase the power. So when we looked at the dispersion and estimation plots between the groups, there was actually very little difference between them. And because Callisto maintained a more diverse gene set for downstream analysis and it had a slightly improved fit versus string tie, we used this data set going forward. And whilst we're on the PCA plots, I should probably draw attention here to that we have a consistent separation of the samples by genotype. In particular, this presence or absence of the SHRSP genome. So these A's here are WKYs, and they sit very separately to the SHRSPs and then the 2 congenic strains, the F being the SHRSP background and the D being the WKY background strain. So as I said, we compared the parental strain as well as each chromosome 14 to its background. And we seemed to have induced a greater effect in the WKY by breeding in that SHRSP chromosome than we did in the SHRSP by breeding in a similar region from the WKY. But across these 3 comparisons, there are 31 genes that are shared across the 3. And when we look at these more specifically, the ones that are boxed here are the ones that are on chromosome 14. We seem to find that the genes tend to go in the same direction between the original parental strains and the WKY background congenic. And then this is the opposite way around in the SHRSP background congenic, which again is nice to see, is probably what would be expected. So if we do an over-representation analysis of those 31 genes that are shared between the groups, we see that these genes have a potential role in transcriptional regulation, but also in regulation of signaling and transduction pathways that are commonly linked to cardiovascular disease and in particular hypertrophic responses. So this P13 ACT signaling pathway and the ECM proteins. We used ingenuity pathway analysis to look at the whole data set in each of the comparison groups. And then when we compared this, we found that mitochondrial dysfunction is increased whilst oxidative phosphorylation is decreased in these WKY background congenics and in the original parental comparisons. We also see key genes, key disease pathways such as cardiac fibrosis and hypertrophy, and there seems to be differences in pathways suggesting transcription is affected in these models. So we then employed this differential transcript usage to see if genes that perhaps there were genes that perhaps are not differentially regulated at the gene level, but when we look into what transcript is underlying that expression, it is different between the models. And what we found is that there was a fair degree of overlap between the genes that were implicated through differential gene expression and differential transcript usage, but there was an additional 200 or so genes that were implicated only by looking at this differential transcript usage. And again, if we do an enrichment analysis of these genes, they seem to be involved in disease functions, including dilated cardiomyopathy, calcium signaling, and fibrosis. So it's probably important that we have a look. So we also looked at the tox functions on IPA in those genes with differential transcript usage, and they seem to be enriched in pathways that culminate in left ventricular dysfunction. And again, these animals, they're at gestational day 18. So this is preceding the development of any functional differences. And the genes that are involved in this pathway I've just shown up here on from a string network. And we see that there's genes such as the natriuretic peptides, integrin receptors, and various ECM proteins. So there might be something in there that's worth a bit more of a look. So the SHRSP genome is associated with differential expression of genes involved in cardiac metabolism and energy usage, and this is potentially priming hearts that become pathological later in life. Transcript usage in the heart is influenced by the background and congenic region, and this alters alternative splicing events in genes that are associated with left ventricular hypertrophy. Pipeline of analysis is probably an important factor to consider. Are we looking to capture genes with low expression? Are they going to be important, or are we okay maybe reducing? So the string tie data had quite a lot of noise in it, so it's quite difficult to find those differentially expressed genes from that really messy data. I spent some time in May at the rat genome database, and they were teaching me all about variant calling and SNPs and indel from alignments both to the SHRSP genome from Peter Doris, but also the Brown Norway. And I'm currently going through that data to see if there are any sort of genes that have SNPs and indels that might be affecting gene expression in our GD18s, but unfortunately that wasn't ready for presentation today. And we're also doing a detailed analysis of genes in the key pathways that I was talking about earlier. So just a quick thank you to the funders, British Heart Foundation, for all of their support during my PhD, the other members of the lab group and my supervisors, the staff at the CVRU at Glasgow who look after the animals, and a big thank you to the guys at the Rat Genome Database for hosting me back in May. It was a great experience, and I am very sad that I couldn't present that data today, but maybe next year. And I'll take any questions.